Okay, from the formation of gametes, we're now going to talk about Mendel's first law. So, as we saw in the formation of gametes, we got a brief reflection and review of the steps of meiosis, and specifically in relating to how gametes are formed. Mendel, it turned out, figured out what was happening in meiosis without ever seeing it. You have to remember during the 19th century, microscopes were just coming into use in biology. So we had looked at some cells, but we had not looked at any of the things going on inside of cells. However, Mendel was able to extrapolate from his data essentially the basics of meiosis. Now, unfortunately, in this section, they haven't really spelled out what his first law is. So I'm going to start off by that. And what Mendel was able to surmise after he did all of these P's crossings was first, that each individual has two factors, what we call alleles, for each trait. In essence, he surmised that the parents are diploid. Because that's what it means to be diploid. You have two copies of each one of your genes. Each of those may be the same allele or they may be different alleles, but you have two copies. Number two, these factors are going to segregate to make gametes. So these factors, i.e. alleles, separate to form gametes. Matter of fact, this is what we just saw. We just saw that in meiosis 1. We took a diploid organism and created a haploid organism. And then in meiosis 2, we took that haploid and divided it again, separating the sister chromatids. Remember, the first division separates the homologous pairs. So now you only have one copy, but each of those homologous pairs has two sister chromatids. And so we then separate those so that we get our 4 cells, each with one copy of that allele. So he never saw this under a microscope like we can today. We can see our chromosomes separating. But he was able to determine that they must go their separate paths in order for recombination to occur. After this, we're going to note that each gamete has one copy. So as we just pointed out, by the time we get done with meiosis 2, each of those sperm cells will only have one copy of that gene. Each of those eggs will only have one copy of that egg. So, in order to become adults again, to form the next generation, we need to get back to being diploid. So the last thing that he was able to surmise was gametes recombine during fertilization to create a new individual. And that individual is back to being diploid, back where we started. So we have a sperm cell, which has one copy. We have an egg, which has one copy. They will reunite through fertilization, and now we have a diploid cell again. This first cell is what you call the zygote. The zygote will then undergo mitosis, to create many more cells. Remember, the role of mitosis is twofold. One is for growth. We undergo mitosis to create a multicellular organism. And then later on, we have to replace cells as they go bad. So these are the four principles of segregation. And you can see how they relate directly to meiosis and the human life cycle.